Hello and welcome to Module 6, Lecture 1, where we're going to be looking at Chapter 8, entitled Bacterial Genetics. And this is good news, everyone. We are now beyond the halfway mark through the term. And from here forward, we're going to be learning about new materials rather than focusing our time and energy on concepts of cell biology that you have or should have learned elsewhere in other classes. Last chapter, we considered DNA replication and the central dogma of molecular biology. That is, taking DNA and making a gene product. In this chapter, we're going to expand further to consider bacterial genetics. Specifically, we'll consider terms like vertical transfer and horizontal transfer of genes. We're going to examine the three types of horizontal transfer: transformation, transduction, and conjugation. We're going to consider gene mutations. We're going to look at spontaneous mutations, the single base mutations, frameshift mutations, transposons, and then we'll move on to induced mutations, that is, mutations in DNA resulting from exposure to a chemical or to some form of radiation. Ultimately, we'll review the different sources of genetic material in bacteria, including chromosomes, plasmids, and the bacteriophage. To begin this chapter, let's consider three definitions of genotype, phenotype, and natural selection. The genotype of an organism is the genetic makeup of an organism, and it includes all of the organism's DNA, the information that codes for the characteristics of the organism. In the case of bacteria, we'll see that most of the genotype of the bacteria is found on a circular chromosome in the nucleus of the cell, but we're also going to see plasmids add to the genotype of a bacterium. Plasmids are an extrachromosomal source of DNA. As we look at bacteria in this module, we're going to consider various ways by which a bacterium's genome may change. We'll see that bacteria have a much more fluid genetic makeup than organisms like us as humans. I find in this chapter, we see that bacteria are just beautifully complicated. We also have phenotype. In contrast to the genotype, the phenotype is the manifestation of a given genotype. It's the visual or the physiological properties in an organism, the way the genotype is expressed. And then finally, with natural selection. Natural selection is something we'll consider this chapter, especially as we look at bacterial adaptation to certain conditions like antibiotic resistance. In essence, natural selection is the process by which organisms better adapted to a particular environment tend to survive and produce more offspring than those that aren't well adapted to a particular environment. Bacteria have a unique ability, well beyond our own human capabilities, to undergo genetic change. And I don't want to mislead you. Yes, bacteria have the ability to undergo normal genetic change, just like us, with mutations that arise in their genome spontaneously, a change in the genome that's passed along to future generations. Genetic change like that is easy to study and track, given bacteria reproduce asexually through a process called binary fission. But what I'm meaning, bacteria have a unique ability to change their genome within their generation, and they have three ways to do so. They can modify their genome via transformation, via transduction, as well as conjugation. And we call this ability to undergo genetic change within their lifetime. We call that horizontal gene transfer, or horizontal change, as compared to changes that are passed down from one generation to the next, which we call vertical gene transfer. To introduce you to the first method of gene transfer, I want to introduce you to a scientist from the 1920s. His name was Frederick Griffith. He was an English microbiologist. And Griffith was looking to understand what comprised genetic material. These were studies that were very popular at the time. Was our genetic material protein? Was it RNA? Was it DNA? And by doing some studies, he was looking at pneumonia following the flu pandemic in the early 1900s. So, of course, the flu pandemic was a result of a virus, but patients who had that virus often were more susceptible to pneumonia as a result of a bacterial infection. So he was looking at the bacterium called Streptococcus pneumoniae, and specifically, he identified two different colony types. A colony type he called type 2R because it was rough looking on the surface texture of the bacterial colony was very rough looking. And then he had a type 3S bacterial colony with a smooth, shiny looking surface texture. Now, the smooth textured bacterial colony was made up of capsuled bacteria, whereas the rough looking colony did not have capsuled bacteria. They were unencapsulated. And so Griffith did some experiments to understand the seriousness of these two visually different colonies as they contributed to pneumonia. And he found mice injected with the type 2R bacteria, the rough-looking, unencapsulated colony bacteria, didn't develop pneumonia. pneumonia. So those mice injected with that type 2R didn't develop pneumonia. We call that avirulent, whereas the mice that were injected with the smooth-looking encapsulated type 3S bacteria, those mice went on to develop pneumonia and they died. And we call this type of bacteria a virulent bacteria. So Griffith concluded that the capsule shielding the bacterium on the type 3S bacteria prevented the immune system of these mice from responding, resulting in this more severe infection that led to death. Now, you're probably wondering where this ties or how this ties to the idea of transformation. Just let me talk through the next steps of Griffith's experiment and I'll get you there. So Griffith then decided he was going to heat kill both bacterial colony types. And again, he injected mice with the type 2R unencapsulated bacteria, as well as the type 3 encapsulated bacteria. And neither mouse developed pneumonia, which indicated to him that heat had killed something in those encapsulated type 3S bacteria, preventing those bacteria from causing infection as they had in their living form. Next, Griffith mixed some bacteria together. He took heat-killed type 3S bacteria that were encapsulated and live type 2R bacteria, those unencapsulated bacteria. And so he mixed those and injected them into mice. And he found this combination of bacteria led to pneumonia and the mice died. How is it that heat-killed type 3S bacteria on their own couldn't kill mice? How is it that heat-killed type 3 bacteria on their own couldn't kill mice? But when mixed with live type 2R, unencapsulated bacteria, living type 2R, those heat-killed type 3S encapsulated bacteria could result in pneumonia and death. Paradoxically, analysis of their blood revealed large numbers of living type 3S encapsulated bacteria. And as far as could be determined, these type 3S bacteria were identical to the type 3S strain from which the heat-killed cells had been made. Griffith concluded that heat-killed type 3S encapsulated bacteria somehow converted live avirulent type 2R cells into virulent type 3S cells, and he called this phenomenon transformation. He further suggested that the transforming principle might be some part of these type 3S cells, and of course he didn't know what that transforming principle was. So that story aside, this is the first topic of horizontal gene transfer, the idea of transformation. Now, in transformation, a bacterial cell, we'll call it a recipient cell, can take up loose genetic information or genetic material from the environment. We call it donor genetic material, where it can go on to be incorporated into a recipient cell's genome. And that's what happened here. I've made it sound very easy, but it isn't quite as easy as I've made it sound. Horizontal gene transfer by transformation, the idea of one bacterium picking up loose genetic information from the environment. Cells that have the ability to take up genetic material like DNA from the environment, they need to be competent, which results from alterations in the cell wall and cell membrane, allowing DNA to enter the cell. Some cells have a natural capability to be competent. Some cells we can treat with chemicals for that, but some cells have a natural capability, and this was what Griffith found. The cells that tend to be naturally competent, Streptococcus, Haemophilus, Neisseria, Bacillus, Staphylococcus, and Pseudomonas. And so this is our first example of horizontal gene transfer, the ability for one cell to release its genetic material into the environment. It's lysed open, it's burst open, it's been damaged. We have genetic material floating around in the environment by which a recipient cell can pick up if they are competent and incorporate that information into their genome. And that's exactly what happened with those type 2R bacteria. They picked up type 3S genetic material, making them virulent and leading to the death of those mice as they developed pneumonia. The next method of horizontal gene transfer is that of transduction. And to discuss transduction, we first need to talk about the bacteriophage, a bacterial virus that acts as a vector to transfer DNA from one bacterium to another. Now, to understand the bacteriophage's contribution to transduction, I need you to know a little bit more about what we call the lytic pathway of bacterial infection. As we talked about earlier in the term, the bacteriophage, or phage for short, is a virus that infects bacteria. It's made up of a protein capsid holding genetic material, and it has a tail that's used to inject nucleic acid into the host bacterium. And when we talk of transduction, we talk of lytic infections and lysogenic infections. I'm going to discuss the lytic cycle first as it pertains to generalized transduction, and then I'll go ahead and I'll cover the idea of lysogenic infections in a few minutes when we consider specialized transduction. In a lytic cycle, a bacteriophage recognizes the host bacterium and attaches. We call that process adsorption. It recognizes specific cell surface receptors on the bacterium and uses these receptors to attach to the bacterium. Then it uses its tail fibers together with a chemical called lysozyme, degrades the peptidoglycan cell wall to create a hollow tube through the bacterial cell wall, where it then injects its nucleic acid into the bacterium. And from here, the viral genome overtakes the bacterial machinery used in gene expression. It destroys the bacterial host genome and uses that machinery in the cell to replicate its own genome, making proteins for protein capsid, replicating its own nucleic acid. We call this the synthesis phase of the lytic cycle. Thereafter, once all the parts of the bacteriophage have been made, bacteriophages are synthesized. They are made, the actual bacteriophage. Nucleic acid is placed into capsids, the tail fibers are assembled, and suddenly there's just no space left in the host cell, and the host cell lyses. And that results in these assembled bacteriophage bursting out into the environment where they can go on and infect new bacterial cells. And so this is the lytic phase of a viral infection for bacteria. Now, why is all this important? Well, to understand transduction, we must know how the lytic cycle works, because what happens in transduction is that a little mistake in the process of transduction unfolds, specifically in the process of generalized transduction. As the bacteriophage packages up some nucleic acid, during the assembly process, it accidentally packages up a little piece of host DNA. And then with the lysis of the phage that contains that little piece of host DNA, the bacteriophage can go on to infect another bacterial cell, releasing donor DNA into a new host cell. And this sums up the process of generalized transduction. I'd like to go ahead and draw it out for you. So let's imagine we have a bacterial cell here that has a genome in green. So it's circular chromosome in green. And then we have a bacteriophage that comes in and attaches itself. It adsorbs to the bacterial cell wall as it recognizes some cell surface receptors. And what happens then is it will use its tail fibers together with lysozyme to penetrate through the cell wall, leading to the release of, nucleic acid. So that viral nucleic acid in red. Now, what's going to happen is that viral DNA is going to go ahead and lend to the destruction of the host cell DNA. So we see that being destroyed, and we still see our viral DNA in red. Gradually, we are going to make new capsids. The viral DNA takes over the machinery of the cell. We make new capsids, we replicate viral DNA, and we had a few little pieces of host cell DNA there. At some point, that bacterial cell will get so big, it will ultimately lyse open. So we have our bacteriophage now assembled and mature, and it's so big that it wants to lyse open. As one of those bacteriophage were assembled, we see here a little mistake. We see bacteriophage that has a piece of bacterial DNA enclosed. That's going to burst open. We'll say lysis. And now we have new bacteriophage hanging out in the environment. And specifically, we want to focus on this one, which is going to come in and now infect a new host cell. So this was host cell number 1. Now we are at host cell number 2. And so we see bacteriophage come in, attach, and gradually that new host cell number 2 will incorporate that piece of host cell number 1 DNA. So thus, we have horizontal gene transfer. Now, in addition to generalized transduction, the bacteriophage can also facilitate the process of specialized transduction. But before we dig in too deeply to specialized transduction, let's discuss the lysogenic cycle of viral infection. We talked about the lytic pattern of infection in generalized transduction, and that's well and fine, but some bacteriophage have a modified replication cycle called a lysogenic infection, and it's a little different than the lytic cycle. In a lysogenic infection, the bacteriophage attaches and inserts its nucleic acid into the bacterial host cell. However, unlike a lytic cell, the host cell's DNA isn't destroyed. The phage genome does not immediately assume control of host cell machinery. Instead, the virus remains inactive, and we refer to it as a prophage. It inserts itself into the DNA of the bacterium, becoming a physical part of the bacterial chromosome. And then every time the cell replicates its infected chromosome, the prophage is also replicated, such that all the daughter cells of a lysogenic cell are thus infected with virus. Before we move on to talk about specialized transduction, you might be wondering what the point of the lysogenic infection might be. Well, from the bacterial perspective, sometimes a lysogenic infection can change the phenotype of a bacterium from a harmless bacteria into a more pathogenic form of a bacterium. A bit of viral DNA has the ability to do this, and in fact, bacteriophage genes can be responsible for toxins as well as other disease-causing proteins found in bacteria that cause diseases like diphtheria, cholera, and rheumatic fever. So back to our specialized transduction example. Time has gone by, the original bacterium is replicated, and there's a colony of cells that have viral DNA incorporated into the bacterial chromosome. At some point, the prophage is going to be excised from the chromosome. Oftentimes, this results from some environmental trigger, some harsh condition, a change of nutrients, a change in space availability, a temperature change, a pH change, moisture change. Some kind of harsh condition lends to the prophage being excised. And in this manner, the lytic steps of synthesis, that assembly and release, resume just as we saw earlier. The cell becomes filled with virions and then it lyses. And so the specialized part of this idea, specialized transduction, it occurs as a result of the prophage being excised. And again, it's an error that's made, some kind of a mistake, if you will, that results in specialized transduction. In addition to the prophage being excised from the host cell DNA, a little bit of host cell DNA is also included in the excision, such that when the newly packaged bacteriophage goes on to infect a new bacterial cell, it inserts its own nucleic acid as well as a little bit of bacterial DNA from the original host cell. And what results? Well, it's possible that the enzyme in the bacterial cell that helps incorporate genetic material might take up that donor bacterial DNA into the new host chromosome, incorporating the original bacterial DNA into the new bacterium. Thus, another example of horizontal gene transfer. So I'm going to draw this out for you as well, and we'll go back to the drawing pad. Looking at the process of transduction, I've drawn some things out just to make this process go a bit easier in understanding. We are going to start with host cell number 1, by which a bacteriophage is going to attach to those cell surface receptors of the bacterium through that process of adsorption. And we're going to see insertion of the viral genome into the host cell. Now, something a little different is going to happen. Rather than the viral genome taking over the cell machinery of the host cell, we're actually going to see, and I'm going to make this a little neater, we're going to see incorporation of the viral genome into the bacterial chromosome. So it will insert itself. And now our bacterial cell is going to undergo binary fission. And I'm going to draw this out real quick. And in this manner, we will see that with each replication, so from 1 cell to 2 cells to 4, 4 to 8 to 16 to 32 to 64 to 128, etc., each generation will have not only this green bacterial genome in the form of a circular chromosome, but the prophage from that bacteriophage is also going to be inserted, and it will replicate as well. Time goes by and we face some kind of a harsh condition. Now, some of those harsh conditions can be a pH change. We might see a significant temperature change. We could see moisture change. We could see nutrient change. We could see space change. We certainly could see UV light being an issue. Some kind of a harsh scenario will lead to the excision of the prophage. And so now that piece of red is going to pull itself out of a variety of these daughter cells that began with this cell right here. Now, a mistake is made during the excision part, as I mentioned, we are going to see little bits of bacterial genome that are pulled out or excised with that prophage. And that's an accident. That is the mistake. And I'll just make a note here saying this is the mistake. Now we are going to go through what we call the lytic phase. So now we convert to the lytic phase. Up here, we were looking at these first processes or the lysogenic phase. So now we have lytic phase, and we're going to go through the steps we did before. The viral genome is going to dissolve the bacterial genome, and it will give instructions taking over the machinery of the host cell, and we are going to see the production of new components necessary. So here's some tail fibers. We're going to see production of our bacteriophage. Now, in addition to that, we are going to copy with this little mistake in place. And so this is where we have something slightly different than what we did before. Now, when these are all put together in the assembly phase, we certainly could have generalized transduction here. We're not really focused on that at this point. We will focus on the specialized component of things. And so that's where we're going to see viral genome as well as little bits of our bacterial genome here. We will see lysis. That's where we're going to see this burst open. And what we're going to be focused on now is this right here. So we are going to see this come in and begin here. This is now host cell number 2. So I'm denoting that with new colored chromosome, yellow. We still see that bit of mistaken excised piece of bacterial genome together with the viral genome in this bacteriophage is going to go on and dump its material into host cell number 2. And from there, we will see, through some enzymatic processes, we may see incorporation of bacterial DNA from host cell number 1, thus providing us horizontal gene transfer. The third method of horizontal gene transfer is through a process we call conjugation. And unlike the typical donor cells involved in the processes of transformation and transduction, the donor cells involved in conjugation remain alive. And to transfer genetic information, they require physical contact between themselves and the recipient cells. So, how does conjugation work? There are two methods of conjugation: plasmid transfer conjugation and chromosome transfer conjugation. Let's begin our look at conjugation by considering the plasmid transfer conjugation. Conjugation via this manner is mediated by something called pili, something called a conjugation pili, a sex pili, thin protein-based tubes that extend from the surface of one cell, the donor cell, and connect to the surface of another cell, the recipient cell. The gene encoding for conjugation pili is located on a plasmid called the F plasmid. Remember, plasmids are small, circular, double-stranded sources of extra chromosomal DNA. And these F plasmids stand for fertility plasmids. Cells that contain an F plasmid are called F plus cells, and they serve as donors during conjugation. Recipient cells are F minus, that is, they lack an F plasmid and therefore have no pili. So the process is pretty straightforward and is shown right here. First, we're going to see that the pilus of the F plus cell contacts the F minus cell and draws the two cells close together. And so we see that happening here. Next, a single strand of the double-stranded F plasmid DNA transfers to the recipient cell. And so we're going to see this process begin here, and we're seeing that transfer. We see a transfer of one of the two strands of the plasmid. And so then the F minus recipient, so that cell here, is going to synthesize a complementary strand of the F plasmid DNA, becoming an F plus cell as well. At the same time, this cell is going to synthesize a new complementary strand of that plasmid DNA. So at the end, what we're going to see is we start with one cell that has a plasmid, one cell that doesn't. This was the F plus, this is the F minus. And through these steps, we are going to see two cells, each having a plasmid and each ending up as F plus cells. Now that is one way of conjugation in this horizontal gene transfer process. We're going to see now the chromosome transfer conjugation process. So this process of chromosome transfer conjugation, the F plasmid does not remain independent in the cytosol of our F plus cell. In fact, we're going to see that the plasmid is going to integrate or incorporate within the bacterial chromosome. And so we see that process here. So that's what happens before this transfer of genetic information takes place. We see a plasmid that's incorporated into the chromosome of the bacterium. And those cells, they are called HFR cells. HFR stands for high frequency of recombination. And they can conjugate with an F minus cell, just like we saw earlier. After the F plasmid has integrated and the HFR and the F minus cells joined by a sex pilus, we're still dealing with a sex pilus, DNA transfer begins. So we're not talking about plasmid transfer necessarily. We are going to see DNA transfer begins at the origin of transfer of the F plasmid. So F plasmid may start this process, but it's going to carry a copy of the donor's chromosome. So this donor bacterium's chromosome is also going to be brought in. And we will see that as that takes place, sometimes what happens is there's this interruption of this transfer of genetic information. Most cases, movement of the cell breaks the F pilus, that intercellular connection between the two cells, before the entire donor chromosome can be transferred. So that whole bacterial cell that's acting as a donor, that HFR, maybe we don't get the whole chromosome copied. And because the recipient receives only a portion of the F plasmid as well, it remains an F minus cell. But the recipient acquires some chromosomal genes from the donor bacterium. And in the end, the recipient is now a cell that contains its own genes as well as some of the donor bacterial genes. So we don't have an F plus, but we have this transfer of chromosomal genes. Now, let's go ahead and move away from horizontal gene transfer. We're going to consider something called spontaneous mutations. And so we're looking more at vertical gene transfer at this point. Spontaneous mutations usually originate as an error in DNA replication, and they become mutations that can be passed along from one generation to the next. For instance, if I were a bacterium that experienced some random mutation in my genome as a result of an error in DNA replication, say I was replicating my DNA, I made an error, I placed a thymine where a guanine should have been, and there weren't the corrective processes in place to make the correction to fix that. I could pass that mutation along to future cell generations in a process called binary fission. And this is the idea of vertical gene transfer. So we have these spontaneous mutations, 3 different types. Spontaneous mutations can affect any part of the genome. And as we'll see, the effects of mutations can vary. If we consider proteins, spontaneous generations may have no effect on an organism, but more likely, a mutation may impair a protein's function. It can stop or it can slow production of a protein. It can overproduce a given protein. We might see a protein becomes completely non-functional. These are just a few examples associated with protein encoding genes, and there are certainly more genetic material than the protein encoding genes. Those are the ones we're going to focus on. There are 2 major types. We have single base substitutions and frameshift mutations. We're going to look at the single base mutations, and there are 3 types here. And then we'll move on to the next slide, which has frameshift mutations. The first mutation type we'll look at is a single base mutation, of which, again, 3 types: silent, missense, and nonsense mutations. And so what are we looking at here? Well, we're looking at a bacterium with a stretch of DNA, and we're getting ready to replicate this. We're getting ready to prepare to undergo binary fission, and so we have to duplicate our DNA. And this might also then be the idea that we then take that new DNA and we are now making amino acids and proteins as a result of transcribing and translating our DNA. So the mistake is made during DNA replication, but then we see some major errors as we go to gene expression. As we prepare for DNA replication, we have our 2 strands here. Remember, they're called leading strands and lagging strands. And we need to make complementary strands that are semi-conservative. In this first example that we see right here, this is what happens normally. We've made no error right here. We have a T, a G, and a T, for which we would make a complementary strand by producing a strand where T pairs with A, G pairs with C, and our T again pairs with A. And this goes on to produce, like if we were then to undergo gene expression, participate in gene expression, this would ultimately go on to produce an amino acid called cysteine. And so we see that right here. And so this is just how things would normally happen. However, these are going to be the examples of the three types of errors we have: silent or synonymous, missense, and nonsense. In a single base mutation, we're going to substitute one of our bases for another by accident. In this next example, if we look at our 5' to 3' direction, TGT, there's an error. We're going to bring in something else, okay? We bring in an A to match T, and we bring in a C to match G, but we accidentally make a mistake and bring in a G instead of an A to match T. And with the help of some enzymes that look for corrections, maybe the other strand is corrected to match the error on the new strand, such now that we have a TGC paired up with ACG. And in this situation, the very best possible mutation scenario, it matches up with the same amino acid. So it codes for the same amino acid. Thus, there's no change in the ultimate amino acid that results. We still have cysteine. And so if you were to go look at our codon list, you would see that UGC is going to code for cysteine as an amino acid, just as UGU did. But we're not going to have it so easy for these two. In the next example, this is an example of a missense mutation. In a missense mutation, we're going to see the production of a new amino acid in place of what should have been laid down for a given codon. We should have TGT complementary to ACA. However, we end up with ACC as the complement to TGT. And some kind of corrective enzyme comes and fixes that 5' to 3' strand to lay down G to replace T. And this changes the codon. And we saw that before. We saw a change in a codon here, but now we're going to see a change in a codon that's going to change the amino acid being coded for. In this case, we have tryptophan instead of cysteine. And so that could change the entire protein that we're making just by one change in an amino acid. Lastly, we have a nonsense mutation. In this substitution, we find the replacement modifies the mRNA codon in such a way that we code for a stop codon, in this case UGA. Rather than code for a mistaken amino acid, we stop the translation process altogether and we have a shortened protein, oftentimes non-functional. And so these three types of single base mutations, they are going to deviate somehow from our original base pairing and creation of a complementary strand. We have silent or synonymous mutations, we have missense mutations, and we have our nonsense mutations. These represent the three types of single base mutations, such that we deviate from our original base pairing. And in the case of missense and nonsense mutations, the mRNA transcript may code for an entirely new amino acid, or it may code for a stop codon, terminating the polypeptide chain before production should be concluded. We call that a truncated protein. We also have another type of spontaneous mutation. These are called frameshift mutations. Unlike single base mutations, frameshift mutations remove or add nucleotide pairs, thus shifting what we call the reading frame, how we read these sets of three, to the left or to the right. And so first, let's go ahead and look at insertions. In this example here from the textbook, in the blue box, shows normal DNA sequencing, as well as the mRNA transcript, which we see here with codons. Geneticists sometimes refer to this as a wild-type organism's genome because this is the DNA and subsequent mRNA without any mistakes. In turn, here we're going to see some mutant genome. Again, we see the DNA as well as the mRNA transcript with codons, little sets of three. And here in yellow, we're going to see that there's a base pair that's been inserted. And we see this here, an A and a T. And so maybe you can imagine what's going to unfold. We've just changed how our codons will read in our mRNA transcript. In fact, we have to add a sequence here, and that's going to throw everything off moving in this direction. So now, instead of a codon number 2 being UAC like we saw up here, now it's AUA. What once was coding for tyrosine is now coding for isoleucine. And if we go to codon number 3, it was originally coding for GUU, but now it's CGU. What once coded for valine is now coding for arginine. You can see this is going to change the entire amino acid sequence beyond the insertion. Our next codon, AAA, originally coded for lysine, but now it codes for a stop codon. So now not only do we have a change in the amino acid sequence beyond the insertion point, but we have a premature stop. And certainly that doesn't happen all the time, but it increases the likelihood that our stop codon will be in a new spot. Let's go ahead and look at an example of a deletion. In this example, again, blue box shows normal DNA sequence as well as the mRNA transcript with the codons. We look here in the purple box and we notice that there's been a removal of a base pair. In this case, the fourth base pair in the sequence of DNA, T and A. As a result, this changes the way the entire frame is read. We still see our first three nucleotides as the same, with C, G, C paired with G, C, C, but the second codon now is going to pair with something new. This will lend to a new amino acid once the mRNA transcript is translated. Our third codon, we're going to see a change as well. Our fourth codon, we're going to see we don't actually have enough information for a fourth codon in our mRNA transcript. We see that this stops prematurely without even a stop codon. So we have a shortened strand of 3 amino acids, arginine, threonine, and leucine, whereas before we had arginine, tyrosine, valine, and lysine. In this module study guide, you're going to get some practice with these. We move from base pair substitutions and insertions and deletions to another concept, that of DNA transposons or transposable elements, sometimes known as jumping genes. Transposable elements are DNA sequences that can transpose or move within the bacterial chromosome, inserting themselves into various locations of the genome, or they may move between the genome and extrachromosomal DNA. They're present not just in bacteria, but in the genomes of all organisms, including ours. They were first discovered by geneticist Barbara McClintock in the 1940s in her study of the colored kernels of maize or corn. The movement of transposable elements from one place to another in the genome has the capacity to disrupt genes and cause mutations, as well as create chromosomal damage. And as it pertains to our study of microbiology, page 211 of our textbook discusses how transposons can lend to the acquisition of antibiotic resistance between different species of bacteria. There are two types of transposons: traditional transposons and retrotransposons. The traditional transposons is a simple cut and paste transposon. The mobile gene encodes for an enzyme called transposase, which takes the gene in question and catalyzes the excision of that gene, integrating it somewhere else in the genome or in some area of extrachromosomal DNA. In contrast, there are retrotransposons, which act more in a copy and paste type of manner. The mobile gene encodes for proteins that are required for their transposition. In this manner, RNA polymerase transcribes the retrotransposon DNA into an mRNA transcript. That mRNA transcript is then translated such that the enzymes needed for transposition are made. And one of those enzymes, called reverse transcriptase, takes the mRNA transcript and reverse transcribes it back into double-stranded DNA copies, such that the copy can be inserted into the genome in a new spot. In this way, we've copied a gene and inserted it elsewhere while also leaving the original gene in its original location. So these are transposons, and they are considered spontaneous mutations as well. We have induced mutations. As we talk about mutations in this lecture, all cells are exposed to various agents called mutagens, which have the potential to damage DNA, cause induced mutations. Some of these agents are natural compounds found in our environment, like UV light, whereas others are man-made pollutants and chemicals in the environment. We're going to look at 4 mutagens we call induced mutagens. The first category of mutagenic chemicals is the base analog, compounds that can substitute for purines or pyrimidines during nucleic acid biosynthesis. For example, our textbook discusses the synthetic chemical 5-bromouracil, a derivative of uracil that behaves as a thymine analog, but with bromine attached to it or substituted in the ring. The presence of bromine increases the sensitivity of a DNA molecule to UV light, which in itself is also mutagenic. And this is an example. This image comes straight from the textbook. And we take a methyl group and we're going to substitute a bromine within that nucleobase. We have alkylating agents, and we're going to see that right here. These are chemicals that donate an alkyl group, like methyl, to amino acid groups in nucleotides. And as with base analogs, base pairing can be altered due to nucleotide substitution, and mutations can result. And so we end up seeing, with alkylating agents, we end up seeing some interaction in these regions, and that's going to create issues as we move forward through DNA replication, gene expression, through that altered nucleotide substitution process. We have intercalating agents. And here's our example of intercalating agents. These are chemical substances that have a similar shape and dimension as purine-pyrimidine pairs, and that allows them to wedge in between base pairs of DNA. That wedging causes base pairs to distort and the DNA strand to unwind a little bit. These changes in DNA structure may affect DNA replication as well as the process of transcription. Deletions and insertions may occur, leading to frameshift mutations in the process of DNA replication, as well as when we consider transcription. We also have radiation. Radiation is another induced or mutagen. We tend to talk about UV light and X-rays when we're considering radiation. Purines like adenine and guanine, and pyrimidines, the thymine and cytosine, they absorb UV light. And radiation is responsible for inducing pyrimidine dimers, specifically thymine dimers. And here's an example of our thymine dimer up here. Thymine dimers, when two thymines are next to one another in a DNA molecule and they absorb UV light, they're very likely to go ahead and end up bonding together, changing the conformation of DNA, which inhibits normal replication as well as transcription. In contrast, when we talk about X-rays, this is an ionizing form of radiation. X-rays are more energetic than UV radiation. Ionizing radiation can pull electrons from otherwise stable atoms, which can directly as well as indirectly affect genetic material. Can alter our purines, it can alter our pyrimidines in DNA, can break up phosphodiester bonds between the sugar-phosphate backbone, X-rays can cause mutations such as deletions in DNA that can ultimately be lethal to a cell. And so these are the 4 types of mutations that we consider. And our last bit of information for this lecture, this is just an overview of kind of what we've looked at up to date. I want to review the 3 sources of genetic material in bacteria, specifically that of circular double-stranded chromosomes, the plasmid, and the bacteriophage. First, we have the chromosome. The chromosome of bacteria is circular, double-stranded. There's generally just one circular chromosome of bacteria. On very, very rare occasions, there might be 2. The chromosome is haploid. There's only one set of genes. And recall, because prokaryotes don't have membrane-bound organelles, genetic information is found in an area of the bacterial cell called the nucleoid. There is no formal nucleus. We have the plasmid. This is our second source of genetic information in a bacterial cell. And as I've mentioned before, the plasmid is a small circular double-stranded DNA molecule distinct from the bacterial cell's chromosomal DNA. We call it extrachromosomal. It isn't part of the genome of bacteria that replicates in association with binary fission. Rather, it replicates independent of the cell. Plasmids aren't necessary for viability, but they tend to lend to virulence, or the ability of an organism to infect the host and cause a disease. They can be exchanged between bacteria through conjugation, as I discussed earlier, and passed from one generation to the next. Bacterial cells may have no plasmids. They may have one plasmid, or they may have multiple plasmids they've garnered from other bacteria of the same or different species, all representing some different activity. There are 4 different types of plasmids that we might come across in a class like this. We have conjugation plasmids, so we've talked about those. These are the plasmids that carry genes necessary for the expression of the sex pili, allowing for conjugation to take place. And we saw this earlier, again, with our F plasmids. We have resistance plasmids, sometimes referred to as R factors or drug resistance plasmids. These type of plasmids tend to carry genes that provide for resistance to some kind of antimicrobial drug, and they're incredibly beneficial for bacteria. They can be transferred between the same species or different species. We have col plasmids or COL plasmids. These plasmids tend to be found with E. coli. They contain genes that code for something called the bacteriocin. These are proteins that can kill other bacteria in one of 2 ways, either by damaging bacterial cell membranes of other bacteria or by degrading the genetic material of other bacteria. So these allow for E. coli to have a competitive advantage. And then finally, we have virulence plasmids, which encode for structures or enzymes or toxins that promote pathogenicity in bacteria. Our very last slide, This is last slide of the lecture, the bacteriophage. It's a source of genetics and bacteria. We're going to revisit it in chapter 13 when we talk about viruses. So you've been given the exposure, the introduction. Recall when we considered transduction, generalized and specialized, we introduced host cell bacteria into a new recipient or new host cell bacterium with the help of the bacteriophage as a vector. And so with that, I will conclude the material covered in Chapter 8 in our study of bacterial genetics. And as always, if you have questions about the material discussed herein, please reach out to me. We can find a convenient time to meet. Meanwhile, make it a great day.